How can one tiny bacterium multiply into millions in just a few hours? It's not magic, it's cell division, the simple but powerful process bacteria use to reproduce and build entire colonies. In this video, you'll learn how bacteria divide, how binary fission works step by step, and how some species reproduce through budding. For multicellular organisms like us, cell division helps us grow or repair tissue. But for bacteria, which are unicellular organisms, division is reproduction itself. Most bacteria reproduce by binary fission. In this asexual process, one parent cell divides to form two identical daughter cells, each containing a full copy of DNA and all major structures. Here's how it unfolds. Step 1. DNA replication begins at a specific site called the origin of replication. The circular chromosome is copied in both directions around the loop. Step 2. As replication continues, the cell elongates. FtsZ, a key protein that guides division, migrates toward the cell's midpoint to prepare the division site. Step 3. The two chromosomes separate and move to opposite poles. FtsZ assembles into a contractile ring, called the Z ring, that marks where division will occur. Step 4. The cell wall begins to fold inward, forming a septum that gradually partitions the cytoplasm into two sections. Step 5. Once the septum is complete, the cell pinches in two. The FtsZ ring disassembles, and two new bacterial cells, each genetically identical to the parent, separate. So far, we've followed the 5 steps of binary fission, from DNA replication to final separation, a fast, efficient process that can double bacterial numbers in minutes. Want to check your progress? Tap the link for a quick practice test and a concise study guide tailored to this lesson. Quiz yourself, skim the highlights, and jump back in. To round out the picture, not all bacteria divide this way. Some use an alternative asexual strategy called budding, which produces a smaller daughter cell from a growth on the parent. In this process, after the genome is replicated, the cell grows unevenly, and a small protrusion, or bud, forms on the parent cell's surface. Next, the bud receives a full copy of DNA along with essential cell contents, and a cross wall forms at its base to separate it from the parent. Finally, the bud breaks free as a smaller daughter cell. It's genetically identical, but will grow to full size before dividing again. In some budding bacteria, such as Hyphomicrobium and Caulobacter, cells also form stalks or hyphae that anchor them to surfaces, while new cells bud off. Compared with binary fission, budding is slower and asymmetrical, but it still produces genetically identical offspring, another way bacteria survive and spread. Importantly, do not confuse bacterial division with mitosis or meiosis in eukaryotes. Prokaryotic division is simpler and faster, and it does not involve spindle fibers or a nuclear envelope. To recap, bacteria reproduce mainly through binary fission and, in some species, budding. Both are efficient asexual processes that allow these microscopic organisms to multiply and thrive almost everywhere on Earth. Ready to quiz yourself with the content covered in this video? Check out the link in the description where you'll find Quizlet content made just for you.